Hi guys! Today we're going to compare some sequences of genes and we need to use two websites. I've sent you both, the NCBI one and the Clustal Omega. So if you open those, we can begin. Thanks. You should have this NCBI screen open and Clustal Omega open. Please have these two screens open to work with. Let's look at the NCBI one and look at this drop-down menu. We're going to come up here and select gene. I want to look at a specific gene. I'm just going to choose hemoglobin. So I'm going to put in HBB, but you could choose any gene that you may find some similarities across species. Then I hit search. Once I get to this screen, I can see that they have selected the Homo sapiens one as one I'd like to look at. I'm going to go down to my search results and I'm going to choose several from here. I want to look at Homo sapiens. I want to look at cattle, chimpanzee, crab-eating macaque, yeah, a rhesus monkey, and let's see here, orangutan. So those are just the ones I chose. You can choose others, it doesn't matter. Whatever species you want to compare. Then I'm going to scroll down to the bottom of that page and it says send to. I'm going to select my clipboard, add to clipboard. Once it's added it, I can find it over here on my clipboard. Clipboard 6 items. Here I am, all of my 6 items on my clipboard. And so now I'm going to look at their DNA sequence for those genes and compare them. So first I'm going to open up the Homo sapiens one. Scroll down, you will see a spot that says FASTA here, right here. Go ahead and select FASTA because I need a certain structure for my sequence. I'm going to copy this. So start at the greater than sign and copy the whole thing. Then I have to go to my Clustal Omega now. Once I've gotten here, I'm not going to look at a protein, I'm going to look at DNA. That's how I'm entering my sequence. Now I'm going to paste this into here. So this was the sequence I just did. I'm going to show you a little trick that'll just make it easier for you later. Go back up to the top of that sequence and you can see where it says Homo sapiens. I want to be able to see that I'm talking about Homo sapiens instead of this number. So I'm going to back it out until it just says NC_Homo sapiens. I'm also going to take out the space between Homo and sapiens just so I can see this better. You'll be able to tell why later. Now I'm going to come down here to the end of my sequence and just hit enter so I can be ready for my next sequence. Now come back over to your NCBI screen, go back one more time. You should be at your clipboard now. Now choose another one. I'm going to choose cattle next. Select that one. Scroll down, find the FASTA sequence. Again, start at the greater than sign, select the whole thing, hit copy, come over again to your Clustal Omega screen. You should be dropped down right underneath that Homo sapiens sequence and paste. Okay, come back up again to where it said the Bos Taurus for the cattle and back out those numbers just so you can know later what you're talking about. And back out that extra space there. Come down here, hit enter. I am ready for my next sequence. I'm going to come back over to my NCBI screen. Hit back again. Choose another one off of my clipboard. I'm just going to choose rhesus monkey now. I'm only going to do 3 right now. You can do as many or as few as you want. Again, I'm going to scroll down to where I get FASTA. Again, I'm going to start with that greater than sign. Select the whole thing, hit copy, come back over to my Clustal Omega, and go ahead and paste that, come back up, make sure I know what I'm talking about. You just have to remember that the macaca mulatta is the rhesus monkey. Okay, now once I have a few on here, I'm not going to worry about entering more right now. I can do as many or as few as I want. I'm going to look at step 2, set your parameters. I want the clustral W and I want character counts. I'm just going to be able to tell how many DNA bases are in each one. Then I'm going to hit submit. It says be notified by email. This is if I've put a lot in. I'm only putting a few in. It's not a big deal. I'm going to hit submit and then just wait patiently. Okay, it did it really fast. And now I can see my alignment. So if you look through this, these are all of the DNA sequences for those 3. I can see that they have shown the cattle first, then humans second, and then my rhesus monkey third. Okay? Looking through here, every time you see this asterisk at the bottom, that means all 3 of them lined up right there. There are sections where all 3 of them line up quite often. And then there are sections where we have a lot less alignment in here, like this. You can also look over at the side and see that character counts. If we go all the way down to the bottom, we can see that the cattle has a total of 1,643 bases for this gene, that humans have 1,608 bases for this gene, and the rhesus monkey has 1,460 bases for this gene. So they vary a little bit, but we can see our similarities and differences here. Okay guys, hopefully you feel super successful now and feel like you have done something amazing with DNA sequences, because you have. Congratulations and have fun. Bye.